Hi viewers, welcome back to one more other lecture session from this channel. So in this video session we will be studying regarding lipids, nucleotides, primary and secondary metabolites in detail. So here what are lipids or how we can define lipids? Lipids are the specific compounds which are water insoluble in nature. Mostly the specific lipid molecules, they are hydrophobic in nature, they won't dissolve in water. Is it clear? So let me write it on the board. Lipids are water insoluble in nature. Water insoluble in nature. Because basically they are hydrophobic. So here, in order to understand the particular concept, let me give you one common example what generally we experience in our daily life. So here if we consider oil droplets and if we pour the oil droplets on the surface of water and if we stir the specific water so accurately, but even though the oil droplets they float on the surface of water but they won't dissolve in water because oil is hydrophobic in nature. And basically the specific lipid, it contains a specific component which is nothing but what? Fatty acid. Lipids contains specific component which is nothing but fatty acid. So here fatty acid, it is the chief component of the particular lipid molecule, and here the specific fatty acid, likewise the amino acid, they possess or they contain a specific functional group which is nothing but what? Carboxylic group. So look at here, carboxylic group. And in addition to the carboxylic group, even the specific fatty acid, even it also contains a specific R group. And here the specific R group which is a part of the fatty acid, it can be of different types. So let me show it here. The particular R group which is a part of the particular fatty acid, it is of different types, and these include methyl group, or it can be ethyl group, or even it can be hydrocarbon chain. Is it clear? The specific fatty acid which is a component of the lipid, it contains a specific functional group which is nothing but carboxylic group. In addition to the carboxylic group, even the particular fatty acid, it contains an R group. And here the specific R group, it can be of different types. It can be methyl group or ethyl group, or even it can be hydrocarbon chain. So here in case of the hydrocarbon chain, here N indicates the number of carbon atoms. Is it clear? And here the range of carbon atoms in case of the hydrocarbon chain, it ranges around C10 to C25. Is it clear? So it is the range of carbon atoms in case of the hydrocarbon chain. So here we can classify the specific fatty acids simply basing on the number of carbon atoms in case of the hydrocarbon chain. So look at here. So here if a specific fatty acid which is having the hydrocarbon chain, if the number of carbon atoms in case of the hydrocarbon chain, if it is 16, then the specific fatty acid, here it is termed as what? Palmitic acid. So look at here. So here it is nothing but what? Palmitic acid, right? Palmitic acid. Then in the same way, if a specific fatty acid which is having the hydrocarbon chain, if the number of carbon atoms in case of the hydrocarbon chain, if it is 18, then the specific fatty acid, here it is termed as what? Stearic acid. So look at here. Stearic acid. Stearic acid, right? Then in the same way, if a specific fatty acid which is having the hydrocarbon chain, if the number of carbon atoms in case of the hydrocarbon chain, if it is 20, then the specific fatty acid, here it is termed as what? Arachidic acid. Arachidic acid. And in addition to this, here we can classify the particular fatty acid simply basing on melting temperature and as well as presence or absence of double bonds. Is it clear? So here we can classify the specific fatty acids simply basing on the number of carbon atoms in case of the hydrocarbon chain, and in addition to this, even we can classify the specific fatty acids simply basing on melting temperature and as well as presence or absence of double bonds. So if you look at here, easily you can understand. So let me arrange the things here. So here, as I've already mentioned you, we can classify the particular fatty acids into two different types simply basing on melting temperature and as well as presence or absence of double bonds. So here basically the particular fatty acids, they are of two different types, and these include saturated fatty acid and the other one is unsaturated fatty acid. Saturated fatty acid and the other one is unsaturated fatty acid. Unsaturated fatty acid, right? So at first, if we consider saturated fatty acids, so what are saturated fatty acids? So here in case of saturated fatty acids, there are no double bonds. Is it clear? In case of saturated fatty acids, no double bonds are present. No double bonds are present. Then in the same way, in case of saturated fatty acids, melting temperature is very higher. Melting temperature is higher in case of saturated fatty acids. So here, what are the various saturated fatty acids here? Some of the examples for saturated fatty acids, they include lauric acid. Is it clear? So let me write it on the board. Lauric acid. Lauric acid, myristic acid, and the other one is palmitic acid, stearic acid, and the other one is arachidic acid. Arachidic acid. So here all these are nothing but what? Saturated fatty acids. Then in the same way, if we consider unsaturated fatty acids, in case of unsaturated fatty acids, double bonds are present. Double bonds are present in between the two successive carbon atoms. Here double bonds are present. Is it clear? And here in case of unsaturated fatty acid, melting temperature is lower. Melting temperature is lower. Then what are the various examples for unsaturated fatty acids here? The various examples for unsaturated fatty acids, they include oleic acid, linoleic acid, linolenic acid, and the other one is arachidonic acid. Arachidonic acid, right? So here arachidic acid, it is an saturated fatty acid, but whereas arachidonic acid, it is an unsaturated fatty acid. And as I have already mentioned you, unsaturated fatty acids, they possess or they contain double bonds, but here the number of double bonds in case of unsaturated fatty acid, it won't remain similar. It remains vary from one particular unsaturated fatty acid to the other. So here, in order to understand this particular concept, let me explain here. So here in case of oleic acid, here the double bond in between the two successive carbon atoms, here it is present only at the ninth position. Is it clear? The particular oleic acid, it contains double bonds only at the ninth position. Then in the same way, if we consider the specific linoleic acid, in case of linoleic acid, here the double bonds are present at the ninth position and as well as at the 12th position. Ninth position and as well as at the 12th position. And if we consider in case of linolenic acid, here double bonds are present at 9th position, 12th position and even at the 15th position. And if we consider in case of arachidonic acid, double bonds are present at 5th position, 8th position, 11th position and even at the 14th position. Is it clear? In case of unsaturated fatty acids, in case of all the unsaturated fatty acids, double bonds are present. But here the number of double bonds in case of unsaturated fatty acid, it won't remain similar, it remains vary. Is it clear? So here we need to understand one key point. As I've already mentioned you, lipids contain a specific component which is nothing but what? Fatty acid. So in addition to the fatty acid, even the specific lipid, it contains one more other specific component which is nothing but what? Glycerol. Is it clear? Most commonly the specific lipid, it contains both fatty acid and as well as glycerol. And very rarely the specific lipid, it only contains fatty acid. Fatty acid and the other one is glycerol. Glycerol, right? So here, in case of a specific lipid, when both fatty acids and as well as glycerol are present, under such condition, the specific fatty acid, it undergoes esterification with the specific glycerol molecule. Is it clear? In a specific lipid molecule, if both fatty acid and as well as glycerol are present, under such condition, the specific fatty acid, it undergoes esterification with glycerol by taking the advantage of ester bond. So here, in order to understand this particular concept, let me show it on the board. Let me arrange the things here. So as I have already mentioned you, the specific lipid, it contains two specific components like fatty acid and as well as glycerol. If a specific lipid molecule, if it contains both fatty acid and as well as glycerol, under such condition, the specific fatty acid, it undergoes esterification with glycerol molecule. Is it clear? So here, in order to understand, let me explain it here. So here it is nothing but what? Glycerol. Glycerol molecule, right? And here it is nothing but what? Fatty acid. So as I have already mentioned you, the specific fatty acid, it contains a specific functional group which is nothing but what? Carboxylic group. So here the specific carboxylic group, which is a part of fatty acid, it undergoes esterification with glycerol by taking the advantage of ester bond. Ester bond, right? So here it is nothing but what? The fatty acid. So here the specific fatty acid which has carboxylic group, it undergoes esterification with glycerol by taking the advantage of ester bond. So here, basing on the number of fatty acid moieties which undergoes esterification with glycerol, here we can classify the specific lipid molecules into three different types, and these include monoglyceride, diglyceride and as well as triglyceride. So here we are much more concerned regarding the specific triglyceride molecule but not about monoglyceride and as well as diglyceride. So here the specific triglyceride which has three fatty acid moieties, it can be of two different types, and these include fat and as well as oil. Is it clear? Here the particular triglyceride, it can act like a fat, and even the specific triglyceride, it can act like oil. So here you make it a small clarification. When the specific triglyceride, it acts like a fat. So here the specific triglyceride, it acts like fat only when the specific fatty acid which has undergone esterification with glycerol, when it is a saturated fatty acid. Is it clear? If the specific fatty acid which has undergone esterification with glycerol, if it is a saturated fatty acid, under such condition, the specific triglyceride, here it is termed as what? Fat. So here, in order to understand this specific concept, let me give you one common example. So here if we consider coconut oil, right? So here coconut oil, it is composed of 90% of saturated fatty acid, 90% of saturated fatty acid and 9% of unsaturated fatty acid, 9% of unsaturated fatty acid. That means Here the concentration or the proportion of saturated fatty acid, it is comparatively higher in comparison with the unsaturated fatty acid in case of coconut oil. So in general, in case of winter season, the specific coconut oil, it remains in solid form, but whereas in case of summer season, the specific coconut oil, it remains in liquid form. So here, why in case of the winter season, the specific coconut oil, it remains in solid form? Because in case of winter season, the temperature is very lower. Is it clear? In winter, temperature is very low. Is it clear? In case of winter, the temperature is very lower. But here the specific coconut oil, it is composed of saturated fatty acid. Saturated fatty acids, as I have already mentioned you, their melting temperature is very higher. In order to remain in liquid form, the specific coconut oil, it requires very higher temperature. But whereas in case of winter season, the temperature is very lower. So the reason why the specific coconut oil in winter season, it remains in solid form, but whereas in summer season, it remains in liquid form. Is it clear? Then in the same way, when the specific triglyceride, it acts like oil. So here the specific triglyceride, it acts like oil. When the specific fatty acid, which has undergone esterification with the glycerol molecule, when it is an unsaturated fatty acid. Is it clear? So let me arrange the things here. Then in the same way, the specific triglyceride, it can act like oil. So here, when the specific triglyceride, it can act like oil here. When the specific fatty acid, which has undergone esterification with glycerol, when it is an unsaturated fatty acid. Is it clear? When the specific fatty acid, which has undergone esterification with glycerol, when it is an unsaturated fatty acid, under such condition, the specific triglyceride, it can act like oil. So here let us consider one unsaturated fatty acid, which is nothing but arachidonic acid. Arachidonic acid, it is an unsaturated fatty acid which is having 4 double bonds. Is it clear? So once when the unsaturated fatty acid, when it undergoes esterification with glycerol, under such condition the specific triglyceride, it can act like oil. So here, in order to understand this particular concept, let me give you one common example what generally we experience in our daily life. So here if we consider certain edible oils, edible oil like sunflower oil, right? So here sunflower oil, it is composed of higher proportion of unsaturated fatty acid. The concentration of unsaturated fatty acids, it is comparatively higher in comparison with the saturated fatty acids in case of edible oils. Is it clear? Unsaturated fatty acids, they are present at higher proportion in case of edible oils. So as I have already mentioned you, the melting temperature of unsaturated fatty acid, it is very lower. Melting temperature is very lower. So in general, certain edible oils like sunflower oil, they remain in liquid form in summer season, and even the specific edible oils like sunflower oil, they remain in liquid form even in case of the winter season. In case of winter season, the temperature is very lower, but even though the specific sunflower oil, it remains in liquid form because the specific sunflower oil, it is composed of unsaturated fatty acids. Unsaturated fatty acids, their melting temperature is very lower. So the reason why even in case of the winter season, the specific edible oils like sunflower oil, they remain in liquid form, but they won't remain in solid form. Is it clear? Then in the same way, one of the modified form of the lipid molecule, here it is nothing but what? Phospholipid. Is it clear? So here what is phospholipid here? When the specific phosphate moiety, when it successfully attaches to the alcohol, which is nothing but what? Glycerol, under such condition, here phospholipid, it is successfully formed here. So let me show it on the board. Let me arrange the things here. One of the modified form of the lipid is nothing but what? Phospholipid. So here in case of phospholipid, phosphate moiety, it successfully attaches here. So if you look at the structure, easily you can understand. So here it is nothing but what? The methyl group, right? So here, once when the specific phosphate, when it successfully attaches here, now here the specific lipid molecule, it successfully converts into what? Phospholipid. Phospholipid, right? So here the specific phospholipid, they are the structural components of cell membrane. And here, one of the modified form of the phospholipid, here it is nothing but what? Lecithin. Lecithin. Lecithin, it is most commonly present in case of the egg yolk. It is clear? The specific lecithin, it is the specific phospholipid which is present in case of egg yolk. And in addition to the phospholipid, one of the modified form of the phospholipid which is present in case of the nervous tissue, here it is nothing but what? Cerebrophospholipid. Is it clear? One of the modified form of the phospholipid which is present in case of the nervous tissue, here it is nothing but what? Cerebrophospholipid. Cerebrophospholipid. Cerebrophospholipids, they are most commonly present in case of the nervous tissue, right? And then after we will be studying regarding nucleotides. So here what are nucleotides? So let me arrange the things here. And then after we will be studying regarding nucleotides. So here what are nucleotides? So here basically nucleotides are heterocyclic carbon ring compounds. Nucleotides are composed of heterocyclic carbon ring compounds. Right? So these heterocyclic carbon ring compounds, they are termed as what? Nitrogenous bases. So here what are the various nitrogenous bases here? The various nitrogenous bases, they include purines and the other one is pyrimidines. So look at here, nitrogenous bases. Basically they are of two different types and these include purines and the other one is pyrimidines. So here what are purines here? Purines are nothing but adenine and the other one is guanine. Then in the same way, pyrimidines are nothing but thymine, cytosine and the other one is uracil. Is it clear? Then what are nucleotides? All these are the nitrogenous bases. Then what are nucleotides? If we look at the structure of nitrogenous bases, then easily we can understand what are nucleotides. So look at here. If you look at the structure of adenine, is it clear? So here it is nothing but what? Adenine. Then in the same way, if we consider guanine, right? So here it is nothing but what? Guanine. Then in the same way, if we consider thymine, so here what is thymine? If we consider the structure of thymine, so here it is nothing but what? Thymine, right? Thymine. And in the same way, if we consider the structure of cytosine, So here it is nothing but what? Cytosine. In general, when the specific nitrogenous base, when it remains in association with the ribose sugar, here it lead to the formation of what? Here the specific nitrogenous base, when it remains in association with the ribose sugar, here it lead to the formation of nucleoside. So look at here. Let me arrange the things here. When the specific nitrogenous base, when it remains in association with ribose sugar, here it lead to the formation of nucleoside. So here once when the nitrogenous base, when it remains in association with the ribose sugar, here it is nothing but what? Nucleoside. Is it clear? So here what are the various nucleosides here? The various nucleosides include adenosine, guanosine, thymosin, and the other one is cytosine. Is it clear? So here all these are nothing but what? Nucleosides. Then what are nucleotides? So here once when the specific nucleoside, when it remains in association with the phosphate moiety, here it leads to the formation of what? Nucleotide. So here it is nothing but what? Nucleotide. So what are the various nucleotides here? The various nucleotides, they include adenylate acid, guanylic acid. Is it clear? Thymidyllic acid, cytidyllic acid, and the other one is uridylic acid. So here once when all these nucleotides, once when they are capable to aggregate together, here it leads to the formation of what? Nucleic acid. Is it clear? Once when many number of nucleotides, once when they are capable to aggregate together, here it leads to the formation of what? Nucleic acid. So here what are the various nucleic acids here? The various nucleic acids, they include DNA and as well as RNA. So here the specific nucleic acids like DNA and as well as RNA, they act as genetic material. They are responsible for carrying the genetic information from one generation to the another generation. Is it clear? And then after we will be studying regarding primary and secondary metabolites in detail. So what are primary and secondary metabolites? And then after we will be studying regarding primary and secondary metabolites. So here what are primary and secondary metabolites? So here one of the important aspect in chemistry deals with isolation of thousands of organic compounds in case of the various living organisms. Is it clear? One of the important aspect in chemistry deals with isolation of thousands of organic compounds and even identifying the structure of these organic compounds, and in addition elucidating the various biosynthetic processes which are involved in the biosynthesis of these organic compounds. Is it clear? So in general, one particular living organisms are capable to synthesize primary metabolites. They are capable to synthesize primary metabolites, right? So here what are the various primary metabolites here? The various primary metabolites, they include all the organic compounds like biomolecules. Is it clear? All the organic compounds like amino acids, proteins, carbohydrates, lipids, and as well as nucleic acids, they come under primary metabolites. These specific primary metabolites, they play an important role for the sustenance of life on the surface of the earth. And in addition, in case of certain living organisms like plants and as well as in case of fungi, they are capable to synthesize secondary metabolites, right? Most commonly the particular secondary metabolites, they are restricted to the plant kingdom. So here these specific secondary metabolites which are most commonly observed in case of plants and even in case of fungi, they are having some commercial importance and even they are also useful in case of human welfare. Is it clear? So here what are the various secondary metabolites here? The various secondary metabolites, they include antibiotics, drugs, pigments, gums, spices and even rubber. So here all these are nothing but what? Secondary metabolites, and in addition even the tannins. So all these are nothing. And in addition to this, if we consider the biological importance of these secondary metabolites, how these specific secondary metabolites, they are useful in case of the human welfare, easily we can understand. So if we consider some of the secondary metabolites like pigments. So as we all know, pigments are the specific substances which are responsible for imparting one specific color. So what are the various pigments here? The various pigments include carotenoids and even anthocyanin. So here carotenoids, they are most commonly used in case of the photosynthesis. Then in the same way, if you consider the specific anthocyanin, it is the specific pigment which is responsible for imparting one specific color to the flower. Is it clear? In nature, the particular flowers, they are having various colors like red, blue and as well as pink. It is because of the presence of what? It is because of the presence of a specific pigment which is nothing but what? Anthocyanin. Then after, if we consider alkalides. So here what are alkalides? So here alkalides are heterocyclic carbon ring compounds, and basically the particular alkalides which are having the commercial importance, they include morphine and as well as codeine. Is it clear? Then in the same way, if we consider the terpenes. So here what are terpenes? Terpenes are natural biological natural isoprene units, right? So here the specific terpenes, they are nothing but what? They are the natural isoprene units. So here basing on the number of carbon atoms, here the specific terpenes, they can be either monoterpenes, diterpenes and even the triterpenes. Is it clear? If we consider the specific monoterpenes, in case of monoterpenes, here the carbon atom number is C10. Is it clear? In case of monoterpenes, here the carbon atom number is C10. Then in the same way, if we consider in case of diterpene, the carbon atom number is C20. And in case of triterpene, the carbon atom number is C30. And in case of tetraterpene, the carbon atom number is C40. Is it clear? And if we consider some of the other secondary metabolites like essential oil. So here the essential oil, it includes lemongrass oil, right? Lemongrass oil. And if we consider some of the drugs, so here what are the specific drugs which are most commonly useful in case of the human welfare? Here some of the secondary metabolites which can act like drugs, they include vincristine and the other one is vinblastine. So here the specific vincristine and as well as vinblastine, they are anti-neoplastic drugs. That means they are responsible for the suppression of cancer, and even they are also responsible for suppressing some more other disorders like Hodgkin's disease. And even these specific drugs, they are also responsible for the suppression of some specific blood disorders. Is it clear? And if we consider the polymeric substances, The various polymeric substances, they include rubber, gums, and as well as cellulose. So here all these compounds, they are having the commercial importance. Is it clear? So here this is regarding lipids, nucleotides, primary and secondary metabolites in detail. And in the next upcoming video session, we will be studying regarding biomacromolecules and as well as structure of proteins in detail. So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends. Thank you.